participation living. And I'm Chuck Sandy. The president, Amy Keating, had a schedule conflict today, so you've got little old me. So before we get started, I want to just make a few Protein Society announcements. Let me see if I can advance my slides. Continue. So I want to point out that the annual symposium of the Protein Society is coming up next month, July 7th through 14th. And this year, because of the COVID situation, it will be a virtual symposium. And we really appreciate the efforts of Jean Hardy and her team in putting this symposium together. And I hope that even if you're not a member of the Protein Society, that you'll attend this, register and attend for this meeting. It's a very low cost, affordable meeting, even for Protein Society non-members. We hope you'll join the Protein Society as part of attending the meeting. But this will be an especially good year for this symposium because we're going to have quite a number of award talks, both from last year's symposium, which was canceled. So we'll have the 2020 award winners. We'll have the 2021 award winners, and we'll even have a straggler from 2019. And so these will be wonderful talks in addition to the other many scheduled talks. And we also will have a special plenary session to close this. And this will be our departing editor-in-chief of Protein Science, Brian Matthews, who will be speaking, as well as our new editor-in-chief, John Curian. And so we think this will be a really wonderful way to end this meeting, looking both at the past and the wonderful work that Brian has done over many years as the editor of the journal, and looking forward to what John will bring to the table as we move forward. The two of them are overlapping this year, and it's a good time to submit papers to Protein Science. I plan to submit a paper just within a week there myself. So those of you who are members of the Protein Society should have received an email asking you to vote on council and other folks for the Society. If you didn't get that email, please contact the Protein Society and we'll make sure that you get a ballot. And please do vote. Both important, both for the Society and society at large. So, and I also want to announce that I hope that you'll mark your calendars for next year. July 7th through 10th will be the next annual symposium of the Protein Society, which will be held in person in San Francisco at the Hilton Hotel there. And the chair of the meeting will be Walter Chase, and he's put together a wonderful program committee that are working on the program even as we speak. Obviously, those of you who have never been to San Francisco need to go to San Francisco. A wonderful place to have a meeting, and we'll look forward to that. And with that, let me just turn the program over to our moderators, Vanita Sood and Chris Koth, to begin the actual scientific portion of the webinar today.
Okay, great. Thanks a lot, Chuck. Let me see if I can just share my screen here. And you should be seeing a blank slide at the moment.
Yep.
All right. So, yes, just by way of very brief introduction, I'm Vanita Sood. I'm a Senior Director of Drug Disposition and Design at EMD Serono, which is a business of Merck KGaA, Darmstadt, Germany. And I'm joined by Christopher Koth, who is a Senior Principal Scientist and a Project Team Leader at Genentech, and we are going to be your moderators today. So in addition to being your moderators today, we are both also drug hunters working on small molecule and biological drug discovery, hence the theme of today's webinar. But perhaps most importantly, we are both members of the Protein Society. And I guess I just wanted to say a word around the fact that this webinar is the first time we're showcasing industry protein scientists at a Protein Society webinar. But, you know, if you think about it, all good drug discovery really needs good protein science. So biologics and antibody drugs, obviously, those are proteins. The connection to protein science is obvious. but small molecule drug discovery as well. The vast majority of targets for small molecule drugs are proteins. So clearly there is a lot of resource that industry-based scientists can leverage by being members of the Protein Society, including great content like you're going to see today and at the symposium coming up soon. And the other point I want to make is, for those of you in academia, protein science in industry is really very innovative and cutting edge, and it spans everything from protein engineering, biophysics, structural biology, computational prediction design, everything from molecular dynamics to deep learning and everything in between, biocatalysis, enzyme design, you name it. It's going on in industry at a very high level. And so today, you are going to see some highlights from 3 amazing scientists, and we will introduce Patrick, Franziska, and Dawei in a little more detail just before each of their talks. Right after the scientific portion of today's program, we are going to have a CV workshop. So if you're in academia and you're thinking about making the leap to industry, which I highly recommend, you will definitely want to stick around for this CV workshop. You'll get a lot of tips. And even if you're already in industry and you're just thinking of brushing up your CV, trying to make it, make your CV stand head and shoulders above the rest, you'll definitely get a lot of tips. And that workshop will be run by my co-moderator, Chris, as well as his colleague from Genentech, Jim Kiefer, who's a senior director and principal scientist. Just before we launch into the program, a couple of logistical notes. At the bottom of your Zoom screen, you should see a couple of different boxes. The chat box, if you just want to make a comment to the organizers, feel free, but for your questions, please put them in the Q&A box. And you don't have to wait till the end. The presenters can't hear you, so you should type your questions and feel free to type them in real time. You can also type them at any time, so even after the second presentation or third presentation started, you can ask the first presenter. So bearing that in mind, to make it easier for me and Chris, if you don't mind just putting the name of the presenter or their initials, who you're directing the question to, that would be super helpful. And then of course, if you have questions during the CV workshop for Chris or Jim, you can ask those as well. And Chris and I will be moderating those. If we run out of time, don't worry, any questions that the presenters can't answer live, they will take the time to type up the answers in the chat box. Okay, so I think that is it, and I will stop sharing my screen so that we can get started. I will hand over to Chris to introduce our first speaker of the day.
All right, thank you very much.
The second speaker of the program today, Franziska Seeger. She's originally from Germany, and she's currently a senior computational protein design scientist at Novo Nordisk Seattle location, where she leverages protein engineering and data science to advance biologics drug discovery for metabolic diseases. Prior to this, she was a boundary-defying research scientist bringing computational protein design to the research arm of Amazon, the retailer that we all love to hate. Skills she honed as a postdoctoral fellow in David Baker's lab at the University of Washington. She's an accomplished crystallographer and a protein designer, and she was nominated as a Stat Wunderkind in 2018 on the strength of the advances that she's made in protein-protein interaction and molecular machine design. A fun fact, she was also a German chess champion, but today she's not talking about chess. She's going to tell us about computational design of de novo protein-protein interactions, a data-driven quest. And with that, I will hand over to Franziska.
Thank you so much, Vanita, for the kind introduction. I'm going to go ahead and share my screen here. Can you see my slides?
It says you started sharing, but I don't see a slide.
Okay, let me try this again. We just practiced this, so hang on, maybe I need to get out of presenter mode and then go back in and try to be proactive. Maybe that was not the right mode. Can you see my screen now?
We see your screen, yeah.
Excellent.
We see it.
Okay, will this work?
Yeah.
Excellent. Great. Yeah, thank you so much for the kind introduction. And also, it's very exciting to be talking at a Protein Society webinar. Proteins are my favorite subject of all time, so I'm really excited to be here. And I couldn't have had a better plug than Patrick's previous presentation, so because I'll be talking a lot about computational protein design and how that leads to therapeutic development. And I'm actually using quite a few structures that Patrick solved during his postdoc as input. So this is really great, and it's a small community, which is awesome. So just to jump right in, I know proteins don't need much of an introduction for the protein webinar, but I just wanted to give a quick introduction to what I mean when I talk about protein design. And so I think of protein design as the inverse of the protein structure prediction problem, which is obviously to go from a primary string of amino acids into a three-dimensional structure and predict from the string of amino acids what that three-dimensional structure looks like. Protein design, for which we use the Rosetta software suite, is the inverse of that, which asks the question, if we have a three-dimensional structure of a protein that we want to fulfill a specific function, what protein sequence, or in fact, what protein sequences, because often it's not a one-to-one mapping, can accommodate that request and fold a protein into a specific three-dimensional structure. And the reason why this is interesting in the context of therapeutic protein design is because we really think that novel, or what they call them, alternative scaffolds could be a really attractive middle ground between traditional small molecule and antibody drug discovery. Small molecules are traditionally orally available, can be very specific, but antibodies have like a real advantage in that area. And so we think that smaller proteins modalities kind of like sit in the middle and could combine the benefits of both of these traditional tracks of therapeutics. To kind of jump in a little bit, and I should also preface this, that most of the work I'm actually showing today was done during my postdoctoral work in the Baker Lab several years ago, but the tradition kind of continues, and I will talk a little bit more about what I'm up to at Novo now. But traditionally, really, protein interface design has used a copy-paste mechanism, where you take information from existing structurally defined protein complexes, such as antibody target complexes, and incorporate side chains or entire secondary structure elements into native proteins or other smaller proteins that have benefits in terms of size and potentially manufacturability. And these are just two examples of very early work in the Baker Lab by Sorel Fleischmann, Eric Crocker, and Stephanie Baker that either use disembodied side chains from antibodies against the stem region of the hemagglutinin protein here to incorporate into a smaller native protein to inhibit flu infections, as well as using a natural ligand BIM to incorporate into a native protein that has a 3-helix bundle structure to then inhibit BCL2 signaling. So the next question after this really great work that used this copy-paste mechanism was obviously a technological question that says, can we actually do this without having existing complex and interface information of how a protein could be engaged that we're therapeutically interested in? And so in contrast to the previous talk, which talked about partial agonism, we're really kind of mostly interested in inhibition as like the first stage of solving this proof of principle. So the question here is really, can we design high affinity and specificity protein-protein interactions from scratch if we just have a structurally defined target of interest? And so the story that I'm going to tell you is about a principled attempt to really solve this protein-protein interface design challenge in a principled way. The goal here was to really design a hypothesis-driven experiment that would allow us to correlate the computational metrics that we get out of Rosetta with the experimental binding and stability success, so that we can then in subsequent rounds bias our design technology towards things that work. And in order to make this generalized, we of course couldn't just do this for one molecule, even though I'm mostly going to talk about one molecule, which is interleukin 17 during my talk for time reasons. But we really considered a wide plethora of different targets, mostly for their therapeutic impact, but also for their structural diversity and interest in the community. I just wanted to quickly orient you to the overall workflow that we use in order to go through this, and I will go into further detail for every single one of these steps. So first, of course, we need to identify a target and kind of like prepare that structurally, and there's a lot of expert knowledge that goes into where to target and how to target. So there's like the target identification and preparation step, which is the first one. Then the second step is a docking and complex generation step. So there have been pre-existing mini binder libraries in the Baker Lab, which I will talk about in a little bit, that we then dock against the desired target surface as like an initial complex generation step, which then can be optimized in the next step of Rosetta interface design to really optimize the interactions that this binder makes with the target for perfect shape and chemical complementarity. We can then take the sequences that we get out of Rosetta, the protein sequences, and translate them into DNA sequences for a synthetic DNA chip order that can then be transformed into yeast surface display library. And that can then be interrogated in a high throughput fashion for binding and stability. And using next generation sequencing and the advances that came with that technology, we can then use data analysis to really filter out which designs worked in which type of binding pool and learn for the next generation of designs what to improve and what to focus on. So like this is not really a linear process, but you can think about this as really an iterative cycle that can be started again once we have some more data. In terms of target identification, as I mentioned before, I will mostly talk about the interleukin 17, which was like one of the primary foci of my postdoc. And yeah, there's actually, just to make a little bit of a plug, this work was continued in the Baker Lab after my departure, and you can stay tuned for like a really interesting preprint that is probably coming out pretty soon. Great. So I wanted to talk a little bit about the complex generation method and like why we're really standing on the shoulders of giants when it comes to the libraries that we can use as an input into the de novo protein design, without which I think these methods would not really be possible. And so the technology that really gave rise to the solid foundation on this is the design of non-functional protein blocks of various different shapes and sizes with high molecular and atomic accuracy. So this slide shows macrocycles all the way from like 7 residue proteins all the way to 60 residue proteins that we can design with really high atomic accuracy when we compare the computational model, which you see here in color, to the experimentally determined structures in gray or white. And so the RMSD of the computational model to the experimentally determined structure in this slide ranges from 0.8 to 2.1, which really shows that using Rosetta, we can really design completely de novo structures with fairly high confidence. So these structures and a mini protein library that was originally developed by Gabriel Rocklin, who put these mini binders of about 43 residues through a protease assay to challenge them and quality control them for proper folding and stability, was then used to use several different docking mechanisms, some of which were developed in the Baker Lab and some of which were developed elsewhere, to generate a starting complex that kind of looks like this. And here I'm using the hemagglutinin protein and a 3-helix bundle scaffold as a comparison. And so here we're really looking for like an initial placement that makes sense, which we can then optimize on in the next step, which is Rosetta interface design. And I'm showing you a quick video to orient you on how this process works. So we have the beige binder in the pocket of the IL-17 cytokine that we're interested in using. And we're allowing Rosetta in a Monte Carlo type fashion to change the backbone slightly of the binder and change the identity and rotamers of the side chains that interact with the target. So it's really kind of like a very stochastically driven process in which Rosetta is allowed to make these substitutions and optimizations of the cytokine, of the binder at the cytokine interface to really optimize for complementarity, shape, and chemical interactions. We then get several thousand sequences out of this process, and because we wanted to kind of like generally solve this problem once and for all and kind of figure out which methods work the best of these 4 docking methods that we used, we then compared the data that came out of Rosetta with the data that we got from the ZDoc5 benchmark and scored with Rosetta in order to figure out how are we actually doing in terms of like protein design compared to native protein-protein interactions. And to no big surprise, native protein interactions, which are shown here in a histogram of DDG, in orange, are much better and have like a much wider tail that reaches out all the way to the left to negative 130 Rosetta energy units here than the designs. But overall, kind of like the mean of our delta G for the interfaces that we designed was comparable to natives, which was a huge success for us. And we continued and didn't only assess the delta G of binding, which can be assessed with Rosetta, but also assessed shape complementarity, buried interface surface, hydrogen bonds across the interface, and whether we buried any hydrogen bonds or charges that could potentially harm the energetics of our system later on, etc. The real challenge here is that you can generate a lot of different sequences and computational models in Rosetta. However, the success rate was anticipated to be fairly low. And so the real tug of war challenge here was to cover a wide parameter space of Rosetta metrics, but still optimize for a high probability of binding success so that we would have enough successful events to perform meaningful statistical analysis and correlations. So just to kind of like orient you a little bit to the numbers, we called this experiment benchmark 1 and designed de novo interfaces against not only IL-17, but 7 different targets using 4 different docking methods and like a previously determined single design protocol. And we ordered a chip that was the biggest in Baker Lab history up until that point, which was 250,000 unique sequences. I know everybody who's done antibody libraries or any other kind of like random libraries like is laughing at that diversity, but that was a big chip in terms of computational diversity for us. And we ordered them for experimental testing. And when we received the DNA oligo chip, we transformed these sequences into yeast for yeast surface display, and then interrogated the stability of our designs using a protease challenge assay that tested whether our designs would withstand various different concentrations of protease using as a proxy for stability, and then also assessed binding affinity with binding against a biotinylated target of interest. We then used fluorescence activated cell sorting again to figure out binding, as I mentioned, and this is just like an example plot where we show expression of our binder library on the x-axis and target engagement on the y-axis, and the background control will be shown in red here, and I'm using a 50 picomolar IL-17A sample as an example, and what you're really looking for is a linear correlation in the top right quadrant here that shows that the proteins that you're displaying actually do have binder engagement. And we were very excited to see that, like, just right off the bat with completely de novo scaffolds and de novo interface design, as I've previously shown, we got really strong binding down to 50 picomolar without avidity on yeast surface display, which was a huge success at the time. We Unfortunately, like after deep sequencing analysis of like the different rounds of interrogation of this library for IL-17 binding and also chymotrypsin challenge of protease for stability, we've only got 8 sequences out of 11,000 that really enriched in the last 10 nanomolar with avidity round of IL-17 sorting. So that was a little bit disappointing because we really wanted to get a higher amount of success so that we could really do statistical correlations. But even some really nifty Bayesian statistics approaches weren't really able to identify any correlations between the Rosetta interface metrics and our design successes, because the 8 out of 11,000 was just not a big enough number to really correlate. And unfortunately, not much has changed in like the success rate department. However, I don't really want to minimize that we did get completely de novo IL-17A binders using this method, which is a huge success in the field and had never been done before previously. And we were able to detect those at nanomolar concentrations on like a single clone yeast analysis. However, when we first attempted to make these proteins outside of the yeast context, in which the designs were expressed as a genetic fusion to the AGA2 protein and were likely monomeric on the yeast surface display, we saw that when we compared the scaffold that was used to design these binders compared to the actual binder that now included the IL-17A interface, that there was a shift on size exclusion chromatography, which was indicative and correlated with a self-dimerization of this binder by itself when it was made, or like when it was just in solution without the context of yeast. And so that really highlighted a few different hypotheses, and we were asking the question, is this because the interface is kind of like too big for the scaffold protein? Can we kind of do better in a second iteration if we just take the secondary structure element of like these binders that work and kind of like graft them into slightly larger proteins? Can we do that with just the hotspot residues from the de novo interface that we design? Or can we do that using like a completely different method? And so just to show you these different approaches that we used in our second iteration is to really use backbone placement, side chain placement, and a completely new docking technique that really combined the best out of the two out of the 4 docking techniques that we've used earlier to get at the question, why are these binders binding on yeast but not in the context of purified proteins? And so just to show you again how we approached the sorting of the naive library that we ordered as a second round down to about 50 picomolar without avidity binding on the yeast surface display, we realized that the obvious winner of like these 3 approaches that I just showed you was side chain grafting of the interface that was previously identified with rift docking, which is the currently probably best and most successful docking method that is used in the Baker lab, was the most successful and gave the highest affinity binders. So this interface was grafted into slightly larger scaffolds. These are 60 to 65 residue proteins as compared to 40 to 43 residue proteins. And when exactly the same interface of the 43 residue proteins was grafted or like incorporated into this larger library, we actually yielded 100 nanomolar binders right off the bat from computational protein design without experimental optimization that were actually monomeric in solution, and we could detect binding on SPR. I just wanted to say that like this is not an isolated incident. Like there's a lot of completely de novo scaffolds and binding interfaces that are now being generated in the Baker lab. I specifically worked on small mini binders and peptides against IL-17 for the application in psoriasis, and also peptides against the IL-23 receptor for applications in Crohn's and ulcerative colitis and IBD in general. And that target structure, which was actually solved by the previous speaker, Praktik, and we actually collaborated pretty closely with the Garcia lab at Stanford for these projects and also for the availability of these screening reagents. And these binders are currently being commercialized out of the Baker Lab and Institute for Protein Design for further use in these indications. So I just wanted to give you a quick summary of the story that I just told you, and then also move forward to give a little bit of an overview of where I think the power of these techniques are and where I'm currently taking this. Just a quick summary, we designed a 100 nanomolar IL-17 binder completely from scratch without experimental optimization using directed evolution or other techniques, which was really the first of its kind. Iterative global optimization of like different scaffold sizes and the stabilization of the local modifications that we made to these non-functionalized mini proteins initially in a repeated and iterative fashion proved to be a successful strategy. And as I mentioned earlier, the side chain placement of the initially identified protein interface into a larger scaffold was successful. I didn't show data for this, but the second iteration of the IL-17 binders was monomeric and folded and thermostable, and even showed signaling inhibition in human epithelial cells. I wanted to take a few minutes, if there's enough time, to kind of like give an outlook of protein design and how I think its power can be used in the future, specifically for therapeutic applications. And as I hope I've convinced you with my talk, the de novo protein-protein interface design can certainly identify completely new proteins that have never existed before with a fairly low probability of success, yet success does happen. And as I mentioned, there's a lot of exciting work in the pipeline that comes out of the Baker Lab. And I think one of like the future challenges is really this joint optimization problem of foldedness and functionality at the same time, to really ask the question, can the scaffold that I'm using accommodate the functionality that I'm trying to engineer into it? And so here I just wanted to give you a plug of a preprint that was released by Doug Tischer last year, where he uses machine learning and TIA Rosetta to jointly optimize for the incorporation of helical binding motifs and the generation of folded scaffolds at the same time. So there's a lot of new exciting techniques that use machine learning in this domain. And I'm happy to talk about that, but I just don't have enough time to get into that during my main talk here. I think generally solving the challenge of de novo PPI design really requires like a detailed understanding of like this interplay of local and global protein structures and functions, and solving this problem more generally with a higher success rate will really require an iterative approach of computational design, probably machine learning, and experimental testing afterwards. Right now, I'm doing almost exactly what I just showed you on different targets in the rare disease space at Novo Nordisk, and I'm trying to figure out whether building completely new proteins from scratch using a protein-protein interface design platform is a viable alternative to traditional antibody and peptide discovery platforms at Novo Nordisk. With that, I would like to thank all of the people who were involved in this work, especially David Baker and Chris Garcia, and my colleagues in the Baker Lab for their help and inspiration during this time. And I would like to thank you for your attention, and I'm happy to take any questions.
Thank you very much, Franziska. That was a really interesting talk. Just a reminder, if people have questions, they can type them into the Q&A box. I actually have quite a few questions. I will try to narrow it down and stick to just a few. So I was intrigued, just going right back to the very beginning of your talk, where you were comparing the properties of the design proteins to native proteins. That slide flashed by kind of fast, but I was intrigued by the fact that there seemed to be many more hydrogen bonds in native proteins compared to design proteins. And then I was trying to connect that to perhaps what you saw later on, where the protein that you had selected by e-surface display was dimerizing when it was purified. So I think you know where I'm going here. So maybe
Yes.
just ask what
Yes. I think what Vanita is alluding to is that hydrogen bonds give protein-protein interaction specificity over just like being sticky and hydrophobic to self-aggregate or just like bind in a sticky fashion to hydrophobic or greasy patches on other target proteins. And so I would say it's still a challenge. So it's definitely true that native proteins have a lot more cross-interface hydrogen bonds, as well as kind of like intra-interface hydrogen bonds. And we're currently working on that. There's techniques that came out of the Baker Lab with HBNet, which I'm sure you're very familiar with, that could help with like specifically identifying hydrogen bond geometries across the interface that would make it more specific. Unfortunately, when this work was done, it wasn't kind of like at a stage yet where this could be used, but that's definitely something to incorporate in future iterations and pipelines.
Okay, great. And there's a question. Actually, Chris, do you want to ask your question live since you do have the ability to, or would you like me to?
Yeah, sure. And I think it ties into the question you just asked, Vanita. So can you comment on PPI design for membrane interfaces? And I'm thinking more along the lines of things like membrane inserting designs, because I would imagine that if you just design a hydrophobic peptide, it's not that's not going to be a drug. So can you comment on that?
Absolutely, yeah. This is not my work, but work from a former colleague of mine in the Baker Lab. And so I'm not sure whether we've kind of like specifically designed membrane inserting drugs, but there's definitely a paper that describes the computational de novo design of transmembrane proteins. So I can definitely point you to that paper.
Thank you.
And this uses like a modified Rosetta energy function that obviously takes into account membrane potentials over how proteins behave in solution.
Thank you.
Maybe I can just ask a quick follow-up to that, and then I'll come to the actual panelists who are supposed to be asking the questions. But does that get inserted into the membrane if you apply it, or it has to be expressed and then it gets translated like normally? Never mind.
I think it has to get expressed and then it gets inserted, but I'm not 100% sure. It's been a couple of years, so I would have to refresh my memory.
No worries. But let's get to the attendees' questions. So actually, Olamide has a non-scientific question, but a relevant one. They say the structural artwork in your slides is beautiful. What software package was used to generate those figures and animations?
Oh, thank you. So I actually use PyMol, and I use like a specific script that I like that I think was passed down by other people, so I really can't take credit for it, but I'm happy to share that if people want to email me to get it.
Oh, great. Maybe you can put it in the chat later or in the Q&A. Mm-hmm. It's available on GitHub or somewhere. And then Abby is asking, she says, thank you for an intriguing talk, amazing work. Can you expand upon the advantages of de novo protein design versus designing an existing protein? So de novo versus redesign.
Mm-hmm. Yeah, absolutely. So I guess it really, it's hard to answer that question kind of like generally, because I think it always depends on the application. Like for therapeutic purposes, very often, if you have a questionable starting point, for immunogenicity reasons, like just redesigning that is probably not a viable option. So using completely de novo sequences that have never really been seen by the immune system before might be beneficial or have disadvantages. So it really very much depends on the application. There is, I think Patrick alluded to the IL-2 space. There's a recent spin out of the Baker Lab that re-engineered IL-2 for cancer applications. And so there doesn't seem to be a lot of anti-drug immunogenicity for those redesigns, probably because of like very high stability and lack of proteolytic processing that will not allow for proper MHC class 2 presentation. And thus immune system activation. But yeah, it kind of really depends on the application. Sometimes redesign, like in the IL-2 case, is great. Sometimes you want a completely de novo protein.
Yeah, I think that's a really good point, and yeah, Patrick made it as well. What I can say also is the FDA takes that kind of concern extremely seriously, even if it's low risk. It's high risk because it has high downsides.
Yeah.
All right, so we have another question from Patrick. He says, great talk. I want to ask for someone applying for PhD programs soon, what skills could one learn? What skills one could learn to join this field and be a competitive candidate, especially for Baker Lab? Winky face.
Okay, yeah, that's a great question. I guess my background is a completely experimental PhD where I just started kind of like being computationally curious, I call it, during like maybe the last year of my PhD. And something that really helped me to make the transition to a computational scientist is actually online classes such as edX or Coursera. It was super helpful for me how to learn to program in Python, or at least kind of like be Python fluent. Or like literate initially and then become more fluent as we go. It takes a little bit of time to get used to that, especially if you don't have foundational or proper training through an undergraduate course. But I would really encourage everybody to take Introduction to Data Science or Python programming, because I think it will kind of like really enrich your lives intellectually, but also becomes like increasingly important for data analysis. So I think having Bash and Python scripting experience is a huge plus. And I honestly don't know what David's selection criteria are these days to take graduate students or postdocs. So I would defer to him for that.
I think we have to move on to the last talk soon, but I want to squeeze in one more question, Franziska. Did you, and this is a quick one, did you notice any correlation between stability? You did a stability selection with your proteases and expression level. Was there any correlation at all? Did you look at that?
We didn't look at this specifically, but I think Gabe Rockling looked at this in his paper, and I'm not exactly sure whether there was like a strong correlation.
Okay, well, thank you.
I would have to look.
Thank you very much again for a great talk. And with that, I will hand it over back to Chris to introduce our last science speaker of the program.
Great. Thank you, Vanita. Thank you, Franziska. So it's my pleasure to next introduce our